This is Week 6, Cancer Genetics and Genomics, Part 2. For a long time, cancer was diagnosed only after a person reported symptoms to a physician, such as an abdominal bloating, bruising, or fatigue, and then follow-up tests might have included scans or blood tests or maybe biopsy to see whether or not an individual actually had the cancer. Today, however, we have screening programs and we can screen populations who have been identified to be at risk, such as the Ashkenazi Jewish women for BRCA1 and BRCA2, or men who are over the age of 50 for prostate cancer by measuring the prostate-specific antigen. The traditional ways of treating cancer are broad. The oldest method was surgery, where you remove the tumor before it invaded the healthy tissue and spread throughout the bloodstream. The other approaches that were used were chemotherapy and radiation. But what this does was it hits all of the rapidly dividing cells, causing adverse effects when they harm healthy cells. The newer approaches are targeted. They're based on genetic information. We already talked about Gleevec, which is a tyrosine kinase inhibitor. It's highly effective if you match it to a sensitive tumor cell type. For all of these approaches, however, it takes just a few escaped or resistant cancer cells to get out and to leave and start to develop a new tumor. So we are now looking at new approaches to be able to diagnose cancer. The ability to sequence cancer exomes and genomes is providing a new specificity for diagnosing and treating cancer. We have such things as liquid biopsy, which is a less invasive method to monitor a cancer spreading through the body. We have cell-free tumor DNA, ctDNA, which can reveal a response to a treatment. And the genetic information that we glean from this helps physicians to choose appropriate drugs by stratifying their patients. Gene expression profiling can specifically describe the type of cell that turns malignant, which then informs treatment choices. A striking example of what is now recognized as mixed lineage leukemia will be discussed in the next 2 slides. An observation was made that 10% of children with acute lymphoblastic leukemia, ALL, do not respond to the chemotherapy that helps the other 90%, even though all the patients have the same symptoms of fever, fatigue, and bruising. And when one looks at the cells under the microscope, they all look alike. However, gene expression analysis using DNA microarrays, chips, revealed that in 10% of patients who did not respond to standard drugs, the cancerous or malignant cells make too little of about 1,000 proteins and too much of 200 others, compared to the protein production in children who do respond to the standard chemotherapy drugs. Those 10%, after doing the gene expression profiling, were found to have MLL and not ALL. And when this was found out, it was found that they respond to different drugs. If we look at this example of gene expression profiling, we can see that the cancer cells that look alike may be genetically distinct. Now, in this gene expression profiling, we see that there's a leukemias, ALL, MLL, and AML. They differ in gene expression patterns. The columns of the squares represent the DNA from tumor samples, and the rows compare the activities of the particular genes. Red tones indicate higher than normal expression, and blue tones show lower than normal expression. The different patterns indicate distinct cancers, although the cells look alike when you take a look at them under the microscope. Let's now look at chimeric antigen receptor technology. Now remember that the immune system regularly recognizes and destroys malignant cells. But by using this technology, which is referred to as CAR, or chimeric antigen receptor technology, we can improve upon what normally takes place in our bodies. And this technology creates a DNA instructions for a hybrid surface protein on T cells that the body does not normally synthesize. This protein is called the chimeric antigen receptor, or CAR, and it consists of part T cell receptor and part antibody. The receptor part is engineered to guide the T cell to a specific target, such as a malignant cell, and then the antibody part binds to the target and alerts the immune system to respond and to destroy the malignant cells. It's almost like the CAR is a little like a drone, and you can see a picture of this on the next slide of how it works. And this slide is just showing you a diagram basically of how the technology works by producing the T cell and the antibody, and then is how it's put into the human body. CAR utilizes genome editing tools to create the chimeric antigen receptors, and we will be looking at some of those next week. A patient typically receives 1 billion or more altered T cells, and CAR is utilized in multiple cancers and is being tested in others. The first CAR patient was a 65-year-old man with leukemia. He had exhausted all his other options of treatment. He received CAR that was made from his own body, and he recovered in a very short period of time. Now today we utilize what is referred to as universal CAR for those individuals who might not be able to produce their own, and that is referred to as UCAR. Problem deaths have occurred from the use of the chemotherapy that is needed to clear the bone marrow of any malignant cells prior to doing the CAR treatment. Living with cancer entails not just diagnosis and treatment, but also the possibility of recurrence of the disease. This is why patients are checked frequently, even if they feel well, and even if years have elapsed since their first illness. We look at a technique called liquid biopsy. It monitors the cancer recurrence and response to treatment. The cancerous tumors shed DNA into the bloodstream, which can be used to monitor disease and even detect disease in people who do not have the symptoms. I would like you to read this article from the National Human Genome Research Institute on the liquid biopsy. Your discussion for the week is actually on this topic, and there are specific questions which I would like you to submit. On the next slide, you will see the link for the YouTube video for liquid biopsy. This is the link for the YouTube video on liquid biopsy. Next week, we will be looking at next gene sequencing, NGS, which is more common to use today in cancer centers.